Welcome to the second part from this sessions, okay? In the first part, we talk about how we test for different kind of biological molecules or for fats, for carbohydrates, for protein, and also for carbohydrates, we have to test. First test for how we test for starch, and the other test for reducing sugar. Type. Now, after we see these videos and after we see how the food taste is going, we will going to make it in labs. I'm going to tell you some important notes about all these tests. For example, as I told you, when the test is negative, that means the reagent color, what happened to it, to it change or doesn't? Huh?
No, it doesn't change.
I said negative. I said negative. Who said it change?
Oh, negative.
Negative doesn't change.
Yes, I said it. When it's become negative, that means the color doesn't change. But when the taste is positive, that means the color of the reagent is what? Is it changing? Type. Who can tell me what is from these tastes? The only taste that needs heating. It was what?
Reducing sugar.
Yes, Benedict taste for tasting reducing sugar. Excellent. The food, as I told you, you have to make it in a solution. So if we have مثلا powder or we have large pieces, we have to dissolve it in water. Type. Can you tell me which test that we mustn't or we shouldn't add water from the beginning? Fat test. We add the water, but after we adding the ethanol to the sample. Do you get it? Right. By the way, also in starch, sometimes when we test for starch, we can use the sample as it. We can make starch مثلا on a piece of potato or on a piece of onion, okay, without making without grinding them or make it powder or adding water or anything. This is the only test we can make it like this. Okay? Type.
Okay.
And also it's better for others tests if the food the sample like it, we have to what? We're going to make it or prepare it as a solution. But if that is a sample, it was a solution at the beginning, خلاص, we're going to make the test what? Directly. خلاص. But what happens if the sample is solid? We have to make first what? We have crushed it or grind it. We're going to add water and shake it. Then we add the sample. As I told you again, in the case of food, when it becomes in fats, okay, we have to crush it, the food directly with the ethanol without it adding water. And when we get the clear filtrated from it, then we're going to add the water. And if already this sample contains fat, it will give us what?
A milky substance.
Yes, a milky suspensions or a milky emulsion. Do you get it?
Yes.
So this is the important note about all the food tests we see it in the first part of this session. Right. We explained in the class, we finished all the carbohydrates. Okay, but here the importance of carbohydrates here. What is the importance of carbohydrates? Sometimes he asks you for the importance, so you have to memorize this information. Number one, it's a source of energy. For this energy, what your body needed this energy for? If he said the energy comes from respiration, the energy coming from foods, your body needed for many.
Metabolism?
Yes, excellent. طبعا. For the metabolism or metabolic reactions, and also the energy needed to remember for what?
Active transport.
Active transport, because it is the only movement of molecules which is needing or active energy, which is need energy for movement is the ion or the molecules from one place to another through the cell membrane. طبعا protein carriers. We need this energy for active transport to change the shape of the protein carrier, as we took in chapter 3 in moving and out the molecules. Thank you. And also this energy we needed for muscle contractions and manufacturing the large biological molecules and cell divisions. How many types, or we have طبعا the carbohydrates, it's found in many types of many sources of food. The samples for the foods, they are reaching in carbohydrates, like rice, macaroni, breads, potato, honey, and all the sweets and all the cakes. What about if we eat large amount of carbohydrates? What happened to the extra carbohydrates is found in your body? How they store it? Yes, excellent. And they store it in the human in a form of molecule. You have to memorize its name, and in plant another form. And for example, in human animals, they stored as a glycogen. This glycogen is stored in the livers and muscles. But what about any more carbohydrates? It's going to change you to this one. Do you get it?
Yes.
Yes. What about the plant? Here it's going to store it as a starch. By the way, this is a way of comparisons between the type of carbohydrates which is stored in animal cell and in plant cell. In animal cell, we're going to say that it's stored as a glycogen, and the plant cell is going to store it as a starch. And this is found out, for example, this starch is stored in the seeds, in tubers, okay? And also some parts which is used in making the cellulose, which is used to make the cell wall in the plant.
Teacher, only extra carbohydrates are turned into fats? Only extra ones?
Yes, the extra one. Your body خلاص doesn't need it. Your body take the carbohydrates or the energy or glucose they needed for respirations, okay? And they get the energy needed for their own vital activities. The more or the excess of it, it's going to store it as a fat. Here, it's going to store it in the animals in a form of glycogen and in the plants in a form of starch. Okay?
Okay.
We move now to proteins. خلاص, we said that the elements are carbon, hydrogen, oxygen, and most of them they have to contain containing nitrogen, and some of them containing sulfur and phosphorus. What is the basic unit? The basic units here, the question comes when we break down the proteins. What is the basic unit? Is what is amino acids. If two amino acids connected together, we're going to have dipeptide, and the many is polypeptide. But what is the important of proteins is going to for growth and tissue repairs. And also, number 2 here, for formations, protein molecules. What are the examples of these protein molecules, which is found in your body? Hemoglobin, some hormones, enzyme, antibodies, and some protein structures. This is a type of foods which rich in proteins. Okay. One, of course, here there is 2 sources for the protein. We have protein from animals and we have proteins from the plants. Yep. But what about also the excess of proteins? As I told you, the protein you need is going to break down into amino acid. This amino acid is moved from your small intestine during the process of absorption to your blood. Then the assimilation process is happening, and then this amino acid is going to combine again and forming the molecules that your body needs, like the enzyme, antibodies, and so on. Okay, like what happens if we have also excess of protein? There is another process here, is called deamination, and this process is happening inside your liver. What about this amination? This deamination process here, for example, it's breaking down the excess of amino acid in the liver, where it's going to be the nitrogen part here, or the nitrogen containing part is going to change into a substance is called ammonia, NH3. Okay, This NH3 is going to form the urea, and this urea, by the way, which is going to move to the kidney to get out of your body in a form of urine. When it dissolves with water and excess salts, they forming the urine. This urea, by the way, is very toxic substance and harm to stay in your body. It formed in the liver, but your body excreted by the kidney. Okay? Do you get it?
Yes.
So far, any doubts now?
No.
Okay.
No.
And here we talk about the test, and we call that the name of the reagent which used in protein is purity test, and we see this video. And here for the fats, what is the importance of fat? Importance of fat here is a source of stored energy. By the way, now you notice that we have two substances that give us energy. What are the two substances give us energy here in this
Carbohydrates.
Excellent. By the way, the carbohydrates give us much or large amount of energy than the carbohydrates. But we're going to use the energy coming from the carbohydrates faster than the fats. That's why when we reduce the amount of carbohydrates in your body going now to start to breaking down the fats. Okay, so if you want to make a diet yani or regimes, you have to cut the intaking of carbohydrates for your body switching to take the energy from burning of fats. Tamam? But of course, the faster one is the carbohydrates. So that's why when we take carbohydrates, plus your body gets the energy from the carbohydrates intaking and doesn't break down any fats, and the fat is going to store it in your body. طيب. What is its function? Another yani importance, it cover and protect vital organs in your body like the kidney and the heart. And used to make, as you know before, this is previous information, is enter in the making of cell membrane. Yes, and it used for insulation to preserve body heat from forming a layer under the skin, and this is act as insulation to keep your body warm. And the test is called what? How we test for fats? The test is called ethanol emulsion test for the fats. Okay. What happened to the if we eat large amount of fats? I think I explained this before when you were in grade 7. Do you remember when we talked about the respiratory system? Do you remember Mr. Ahmed and the coronary heart disease? Do you remember this?
Mr. Sayed.
Yes.
أحمد وسيد يا مس.
Yes, Ahmed and Sayed. Yes, that's it. Okay, so here we have several factors can lead to this coronary heart disease. Fine. At the beginning, I'm going to explain again what is meant by coronary heart disease. We have, for example, in our heart here, the artery which is supplying the heart muscle with oxygen and glucose to make them pumping or contractions properly is the coronary artery. So here, if we eating a large amount of fat, this is going to block and prevent the passage with the yani is going to restrict the flowing of blood containing also the oxygen for making the respiration to get the energy, and this is leads to heart attack at the end. So here, coronary heart disease is when the coronary arteries that supply the blood to the muscle of the heart get blocked. By the way, there is no any new information here. All of this information we talked before. Okay, so here it's going to be blocked by the layer of fatty materials. This fatty material is going to building up. Okay, and forming here going to be thickness at the wall here of your artery. Okay, like this is because the artery is become what? It's become narrow. And so the blood is what, girls? It's going to be restricted. Yani not enough blood now reach to this artery. And when there is no blood reach to them, this is mean lack of
oxygen.
Oxygen, because the here for the artery is the which is carrying the blood, carrying the oxygen to the heart muscles, and this could lead to a heart attack. Of course, there is many risk factor for these diseases. Okay, like when we مثلا eating large amount of food, like smoking. Okay, all of this is going to lead to narrowing the blood capillary and restricting the flowing of the blood here. Okay, so the blood flow is going to stop, preventing reaching oxygen and glucose to the for respiration, يعني for the coronary artery, and make it just stop.
It makes a clot. لا يعني
Yes,
حتى كده.
it causes clots. Yes, of course it causes clots. These clots happened when بقى يعني when they restricted here the flowing of the blood is going to forming these clots. Okay,
Yes.
as I told you, eating large amount of food containing fats and also smoking here is this from the risks is going to increase this disease, and also we have another risk to being inactive. This lead to high blood pressures, and all of this leads at the end to the fatty deposit, and this goes to the heart attack. Do you get it?
Yes.
Okay. The last test here we're going to take it. This is about I'm going to explain it to you in some day in the school بإذن الله. Okay.
Yeah.
We have to move to the last one is what here is the DNA. As the structure of DNA. Okay, just wait. Here, for example, at the beginning, do you know any information about the DNA? Can you tell me what is the DNA doing or something like this?
It's used to يعني we can
At the
specify
beginning, it's found where? At the beginning. The genetic information or the DNA is found where?
Nucleus.
Yes, excellent, excellent. Yes, so the nucleus here, for every living organism, it has these chromosomes. Okay, but do you know the chromosomes transmitted what? This is the structure of the DNA. Do you know the chromosomes?
Genes.
The chromosomes, it carry what?
Proteins.
No, I'm just saying it. يعني carry what? What is its function? The chromosomes carry what from your dad or something like this?
Genes.
Yes, it carry
Genetic
genes.
material.
Yes, it can transmit what? The characteristics from one generation to another. Here بقى we're going to prove that this chromosome is made up of protein and some biological molecules which we called it nucleic acid. Okay? The nucleic acid here, we give it the abbreviation of DNA. DNA here is very important in, as I told you, in two things happen to you is very, very important. Number one is help us in inheritance. And what else? In protein, making the protein. In protein synthesis. Okay, do you get it now? So here, as I told you, the nucleus here carrying the chromosomes. The chromosomes here, which is can transmit or transfer the characteristics from one generation to another. These chromosomes is made up from two things, from making proteins and the DNA. Now we're going to learn about the DNA and its structure. So here, the structure of DNA, as you can see, it consists of two long strands. Do you see this, the long strands here?
Yes.
yes.
What they doing around each other? They what? It has a name. We can say they are, they collide around each other. And then when they collide around each other, they forming what we called it, you have to write this well, a double helix. Again, so how the double helix is formed, as I told you, the DNA is consist of how many strands? As you see, how many strands here?
Two.
We have two
Two.
strands. Excellent. They collide around each other to forming what we call it a double helix.
Double helix.
Each double helix or each strand, from the DNA has or containing some is called polynucleotides. What's the meaning of nucleotides? Here is the basic unit. Do you remember when we said the protein, the basic unit of protein is amino acids? And also, yes, and also we said the fatty acid and the cholesterol, and we said carbohydrates from glucose or simple sugar. The same here with the DNA. What is the building unit or the basic units for the DNA? We called it single. This is yani we call about it is single what? Nucleotide. Okay, it's called what? Nucleotide. Type. The nucleotide is forming from what, girls? It's forming from nitrogenous base connected to a sugar is called pentose sugar, and also is connected to a phosphate groups. So sometimes in past paper he asked for drawing the nucleotide and mention its structure. Do you get it? So here again, now we talk about the DNA structures. We said how many strands we have here?
Two.
Stay with me, all of you. Yalla, 2 strands. These two strands are they forming
Double helix.
Double helix.
Yes, each strand is making from single units of
Nitrogenous base.
nucleotides. Yes. Yeah. First, you have to say the nucleotides. This nucleotides is consist of 3 main parts. Consist of phosphate groups connected to pentose sugar. Okay. This sugar is why we call it pentose, because it contain 5 here. Okay. And the nitrogenous containing base. Right. Do you remember the nitrogenous base?
Yes.
We have how many nitrogenous base here at DNA, girls?
4.
Yes. We have A for what?
Adenine.
Adenine. And T for thymine. And C for cytosine. And G for guanine. Yes, guanine. Adenine, thymine, cytosine, and guanine. Okay? But they have a role in combining together. According a complementary base pairing. What about the complementary base pairing, girls? You find that always A is it's going to pair with T, and C
With T.
with G. Okay, do you get it?
Yes.
Here the explanation. You're going to study from this page, by the way. So here, what about the nucleic acid? What is the basic element? Is carbon, hydrogen, oxygen, nitrogen, and phosphorus. So here is the DNA is chemical that make our genes and our chromosomes. DNA is material that we inherit from our parents to give us our characteristics. It made up of a smaller unit. Yalla, what is the name of the smaller unit? Is nucleotide. Okay, this nucleotide is consist of or made up of 3 components. This one is called what? Phosphate group. It's called
Phosphate
what?
group.
Phosphate group. And this sugar is called
Bentane.
Code.
no, this is written wrong here. No, it's what? It's
Pentose.
sugar. Okay, pentose sugar, which is consist of 5 carbons. Okay, consists of 5 carbons. And then a nitrogen
Nitrogenous
is
base.
called the nitrogenous base. If he asks you in the past paper and you're going to see how the question come, I'm going to send it in your homework. Describe the structure of DNA. You find that the DNA molecules is consist, يلا you have to say that all the things we explained. يلا, DNA consists of how many strands? How we have
Two strands.
who lie
They're
together.
called double helix.
double helix.
Yes. Each strand of DNA is polynucleotide. Okay. This polynucleotide is consist of the smallest unit is called what? Nucleotide. Yeah. It has many nucleotides. Okay. Together. Type each nucleotide that is made of what?
Phosphate group.
Phosphate group
And nitrogenous
and
base.
nitrogenous base. But how many nitrogenous base we have? We have
Four
four times.
nitrogenous base.
Excellent. We have A for adenine, T for thymine, T for cytosine, and T for
choline.
yes.
Miss?
And the bases. Yes. Any question?
Yes, do we have to memorize the names?
Yes, you have. لا يعني
All of
you're going
these?
no, you're going to memorize the letters. Okay.
Okay.
يعني and you have to learn or you have to memorize that A is called complementary with T. And this is a question come by the way. يعني he give you, for example, in exams, he gives you, for example, here one strand, and he said to you, مثلا, we have here A, we have here C, we have, for example, here T. Tell me here the complementary for the another
A and
one.
T.
So here with A, it will be what I'm going to write here.
T.
No, A with A with T.
T.
Yes, and the C with
G.
T, and the T again here with
A.
and so on. Do you get it? How the question comes here? But he didn't ask about it. But when we repeat it explanation, you're going to memorize with us so easy We have adenine, thymine, cytosine, guanine. The A for adenine, the first letter is A, as you see. The thymine, the first letter is T. Cytosine is the C, and guanine is G. And they are so easy. And the bases always pair up in the same way according to what we call the 8 complementary base pairing rule, which is A with T and C with G. Do you get it?
Yes.
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 The two strands this type. Here, for example, we have the A with T and C with G. The two strands here in the DNA held together by a bond is called, this is a very, very important, is called what? Hydrogen bond.
Hydrogen bond.
It's called hydrogen bond. Okay. So far now, any doubt?
No.
Okay. So, now we finished, alhamdulillah, the biological molecule.